a student at RCC. So if you want to remain anonymous, you can, as you log into Zoom, change your name. You could be SpongeBob SquarePants for all I care, all right? I'm not going to worry about that. And on the Kahoot, if you want to use an alias, you're welcome to do that. So it's my day class, my night class, plus Dr. Sai's classes that will be there. So there will be a lot of people there. Now, yeah?
Did you send the link through the announcement?
Yes, I did. I got the links in the announcement, okay? Don't be late, because once we start Kahoot, I can't let anybody in, okay? So once we start, so if there's not a lot of questions or things like that, then the Kahoot will start and I can't let you in, and then you'll be sad, okay? If you can't make it, all right, it'll be recorded. It takes Zoom about a time and a half to process the recording, and then it will be posted, okay? Now, the review questions on Kahoot came mostly from you. 95% came from students, okay? So, you submitted your questions. Sometimes I tweaked the questions a little bit, and some folks turned in questions that had incorrect answers, which made me very nervous, okay? And then some of the questions I put in, okay? So, is it gonna be exactly like the exam? No. What is the Kahoot gonna try and do? Stimulate your memory, so you know what areas you might need a little more work, okay? Does that make sense? So it's not like, oh, I'll memorize the Kahoot and get 100%. No. This is micro. It's just to review, okay? Do you have any questions about the exam or anything going on? Good. Yeah.
We get those at the bookstore, right? The scan.
Yeah, you should be able to get them at the bookstore. Where did y'all get your scan from? Bookstore? Yeah. How much was it? 75 cents. Give them your ID so you can get a discount. I don't know. I'm always trying. I don't know. Let me know. Oh my goodness. Okay, are you cool? Yeah. The questions on the exam, you're going to get a paper exam, and you can write on it. You can do whatever you need to do on it, and then put your answers on the Scantron, and then that's it. Okay.
We have metrics on?
Yes, metrics, scientific notation, yeah.
And also the disease cards there.
Yeah, there's a section that has disease card questions. Not as heavy as the Quizzy Wizzy, but some. And, you know, it's name, signs, symptoms, system. Yeah. Yeah. Okay. You good? All right. Let's get rolling. We talked about how microbes can, blow up without... We got this far last time, right?
Yeah.
Good. All right. So that means we got to here. So... take some down, pass them around. Okay, there we go. There's some. Right, some more. Here's some more. There we go. There we go. Okay. Extras?
Yep.
No extras? Pass them up.
Pass them up.
Yeah, here, I'll get. Okay. Everybody got one? Okay. Okay. So, let's review, and again, this is Bio 1. Thank you. This, oh, pass it. Okay. Thank you. All right. It's Bio 1. Remember, what is the definition of diffusion? What is the definition? Don't look at the handout right now. What is the definition of diffusion? Movement of a
Concentration gradient.
of something from an area of
High hydration.
to lower concentration. Good. And under that diffusion umbrella, there's osmosis. What is osmosis?
Movement of water.
Movement of water across a semi-permeable membrane. Don't forget that part of the definition. Okay, what is dialysis? Movement of a solute across the semi-permeable membrane. Okay, that's all Bio 1. Okay, so we're going to look at what can cross the membranes of semi-permeable. Okay, we've already talked about all these good things here. Okay, so at the top part of this is diffusion. On the bottom part is osmosis. Go ahead and fill it out according to what you think will be happening. Bless you. Good. Easy peasy, huh? Yeah, good. Okay, so you've got your it figured out. The first one, the top one is pretty easy for diffusion, right? You can do that in your sleep. Okay, now let's reintroduce the terms hypertonic, hypotonic, and isotonic. Okay? For the row that's at the bottom, I want you to label which solutions are hypertonic, hypotonic, and isotonic. When I think you got it, raise your hand. Okay, let's take a look at this. I know it's been a while and this will click in your brain, but that's all right. Okay, it'll just take a moment. All right, so we've got 1, 2, 3, 4. The first box. All right, put 5 up there. Okay, what kind of solution is this? Isotonic. Isotonic. So which way is water going to flow? Both ways. So the net flow, though, is 0, right? Okay, how many of you got that one? Good. Oh, awesome. Okay, let's try another one. Actually, I can do it up here. It's probably easier to see. Okay, let's do the next one. Can you see that better? Yeah. Okay. All right. So we've got this going on, and we've got 2 over here, and we've got like that much. Okay, what's going on here? First of all, which way is water going to flow? It's going to flow that one, okay? Yeah, because why? These are all solutes. Remember, salt, sucks. Okay, wherever the most solutes are, that's the way water's gonna go. Okay, so let's label this. If you labeled this as hypertonic, this whole thing, that's incorrect. Oh my goodness, yes. Why? Because you've got a system of two solutions here. Okay, hypotonic, and this would be hypertonic. Oh, remember that? Okay, now it's making sense. Okay, so when I look at this, and this is my, convoluted brain, I look and I always say, okay, which side has the most solutes? Aha, this one. So this would be hypertonic. That makes this side hypotonic. Okay, once I've figured that out, then I say, okay, salt sucks. Where are the most solutes? Oh, it's here. That's the way water's gonna flow. Okay, fix your cells. Okay, right now, fix your cells. Okay, make sure you got everything down there, because again, this is review, right? Okay, is everything fixed on your handout? Okay, good. So let's look at the next one. Here's box, here's a semi-permeable membrane, and we've got over here into over here. Okay, where's the most solutes? The left. So this part is, the solution is hypertonic. What is this one? Hypotonic. Which way is water going to flow?
To the hypertonic.
To the left, right? Okay. so, yeah, okay? It's going to flow that direction, okay? All right. On the exam, you get a question like this, you may not have a picture. It may give you words. Draw it out, okay? All right? Because I look around a lot of you and I'm concerned. This is part of my concern, is that you should be writing like crazy. Crazy. Did anybody have Virginia McKee-Leone for Miss Virginia for anatomy?
Oh, yeah.
Oh, yeah. She was so good, right? So last semester I sat in while she did neuro, and I had 25 pages of notes from her lecture. Okay? 25 pages. And I looked around the room, and there were people braiding their hair and just staring off into space. And my suspicion was that they were recording it and they were going to do it later. But here's the thing. If you're not writing down everything right now as much as you can, you're missing one opportunity through the material. And remember, we're trying to get many times through the material. So even if you're recording it, you should be taking furious and copious notes, right? Writing down everything. So that way when you go home and listen to the recording, you can add things to it. If you're just saying, I'm just going to come to class and record, you're missing that opportunity. Now, life is about choices. If that's your choice, that's great. But I'm saying from an educational standpoint, the best thing to do is write like crazy. Yeah, I had to go and get another notebook. I mean, she's so cool. And you know what got me is that, like, I said to her after the class, I said, you told them everything that was going to be on the exam. She goes, yeah, and they didn't pay attention. I said, what? She goes, oh, they won't pay attention because they're not into it. They're daydreaming. They're tired, whatever. I said, but you just told them everything. She's like, yeah. You know how she laughs. So anyway, okay, any questions on this? Let's try one you haven't seen before. Okay, and I would be drawing this out, right? Draw. Okay, this is a red blood cell, RBC, and let's do this. Let's say it's 1.5%. I don't want to do that. Let's make it 0 sodium chloride. Okay, in this solution in a red blood cell. Okay, tell me what's hypertonic, hypotonic, and which way water is going to flow. Tell me. Okay, which solution is going to be hypertonic? This one, because what's our concentration for red blood cells? 0.9% sodium chloride. That's normal saline. We've talked about that before. So this solution right here is hypertonic. This one is what? Hypotonic. Which way is water going to flow? Out. What's going to happen to the size of the cell? Shrink. Okay, how many of you got that one? Be proud. It's okay to be like, hey, I got that. Okay? One person. One person got it? That's it? Uh-oh. Okay. All right. You want another one? Okay. Better get this one. Please get this one. Please. Okay, there's a solution there. There's an RBC here. And we've got 0.5. Okay. Now what's going to happen? Okay, which solution is hypertonic? This is right. This is going to be hypertonic. This one will be hypotonic. Which way is water going to flow? In the cell, what's going to happen to that cell? It's going to expand, and it doesn't have a cell wall, so what's it going to do? It's going to lyse, right? How many of you got that one? Now we're talking. Okay, good. Okay, any questions on this before we go on? Good? Okay. Cool. All right. So we've talked about this kind of already, but what can cross a membrane? Water can always cross a membrane, period. That's why we have movement of water to that third space, right? Yeah. Okay? And we can move it back in fairly quickly under certain circumstances. So that's the one thing that can always cross a membrane. Small molecules, things that are lipophilic, big molecules and charged molecules, they don't make it. It's usually molecules that are 3 to 4 atoms large, and that's what can cross, okay? And we're talking passive diffusion. Does that require energy? No. Good. Okay, facilitated diffusion. Okay, this is where solute combines with the transporter protein in the membrane, and this is passive. It's using the power of the concentration gradient to go through here. Okay, we good? Okay, so that's facilitated diffusion. All right, we've already talked about osmosis as movement of what? Water across this semi-permeable membrane. Okay, so we've kind of already done this. We can do this real quick. Okay, try this one. You can look at this one. Which solution is hypertonic? A, which is hypo? B, which way is osmosis? Which way is water going to flow? B to A. Good. How many of you got that one? Good. All right, let's try this one. Which solution is hypertonic? Okay, B. Yeah, if it's got 25% salts, it's only 20% over here. Okay. What solution is hypotonic? Which way is water going to go? A to B, right? Okay, cool. All right, so we've done this. Now, when you talk about passive transport, you're talking about movement of molecules that require no energy. No energy, because why? They're using the potential energy, they're using the energy of that chemical gradient here, okay? Active transport, movement across a membrane that requires what? ATP. Good. ATP. Okay, so there's a famous active transport example, and if you've had anatomy, you should remember this one. What is an example of an active transport system? The sodium-potassium pump. Excellent. Good. Yeah, the sodium potassium pump. If you don't remember that from Bio 1 or anatomy, please review that. Okay? What is happening? Well, you're using your sodium potassium right now. Did anybody see that obituary of that weightlifter that was 20 that died of severe dehydration? Okay? She was dehydrating herself, and oftentimes when they do this, they use something called Lasix or torsemide. What does Lasix do? It dehydrates you. What does it do? It forces all of this fluid into your, I think it's the descending tubule. I have to look that up, but anyway, into the kidney. But along with it comes potassium. And what happens when your potassium levels go down? That's the critical electrolyte, right? Your heart will stop. And that's exactly what happened to her. She's dead at 20. Okay? So, you're using the sodium potassium pump because what do you use it for? For repolarization of the cell membrane so you can move muscles. Yeah. So, make sure to review this sodium potassium pump, okay? So, here we go. Solute binding. Yeah, there we go. And then the phosphate comes off, okay? So, yeah, requiring pumps, integral membrane pumps, okay? The other thing that you see active transport is when you've got group translocation that happens too. Okay, so let's look at how things that are big, large items leak. Okay, this is the cell. This is bulk active transport, okay? What are we seeing here? If it's going out, what is it? Exocytosis. If it's going in, endocytosis, right? And this requires energy to do this, all right? Okay, let's look at some things here. Cytoplasm. The cytoplasm. There's cytosol and cytosol. Okay, so 70 to 80% water. It's a dense jello solution of solutes, okay, like amino acids, proteins, carbohydrates, and soluble organic matter. Serves as a storage for materials, okay? That's what you're going to use this for, okay? So you've got a lot of resources in the cytoplasm, but it's mostly water. Chromosome, okay? The chromosome is a single circular molecule, okay? That's why they don't have histones, all right? Contains all the genetic information. The DNA gets coiled around this protein, and it's called the nucleoid region. It's kind of the apartment for where you keep the DNA, but you don't have a membrane around it, right? Okay. All right. Any questions on this? Did you get all your blanks? Yes. Storage. Storage, yes. Okay, cool. All right, these are great. These are plasmids, okay? Plasmids are small circular double-stranded DNA that are different from the bacterial chromosomes. They're usually smaller, okay? Now, sometimes they're free, like this, but when we do microbial genetics, you're gonna see it in some instances how that plasmid joins up with the bacterial chromosome, okay? Now, there must be an advantage to having these, or it wouldn't be there, okay? You've got 4 million years of evolution. These must be important, okay? When the cell goes through binary fission, not only will it copy the host chromosome, but it will also copy that plasmid, okay? And so they're useful genes from the bacteria's perspective. These are useful genes for its survival. A lot of times you get antibiotic resistance on those plasmids. genes for toxic metal tolerance, other enzymes. Sometimes you get toxins that are coded for in that plasmid. And as we're going to see when we do microbial genetics, bacteria is so promiscuous. Okay? You know about the pili already, right? You can have sticky pili, or you can have some that actually transmit genetic information. This is why microbial antibiotic resistance is so important, because they're transmitting this plasmid with any bacteria that it finds. Pretty skanky, huh? Okay. Used in genetic engineering as vectors, because we can transfer genes cell to cell. Yeah, we do this in the lab all the time. So, a little different way of thinking of another mechanism to transfer genetic information. Yeah?
What is the E for the first bullet?
Useful genes not encoded by the host chromosome. Yeah.
Thank you.
Mm-hmm. Okay, let's do some ribosomes. In eukaryotes, what is the size of the ribosome? ADS. Okay, and what does the S stand for? It's a sedimentation rate. So the way they found it is they took the ribosomes and put it in the little centrifuge, and they saw where they fell out along the tube, and they do at that's at 80. Okay, you have ribosomes, 60% ribosomal RNA and 40% protein. Two subunits, large and small, and this is where protein synthesis happens. In prokaryotes, are the ribosomes larger or smaller? Smaller. How much do they weigh? What's their sediment? What's their S value? 70. Right. Okay. So that's kind of cool. All right. Let's talk about inclusions and granules. Okay. Not all cells, bacterial cells, are going to have inclusions and granules, and it varies. Okay. These bacteria are able to store things when, in case the environmental sources are depleted. Okay, so what are some examples? Well, there's some of the photosynthetic ones. They store starch. Okay, they store iron. They store phosphate. Pay attention to PHB. Okay. That's so they can store glycogen. PHB is poly B hydroxybutyrate. Okay, look that one up. Okay, so you understand PHB. Okay, that's one of the things, and this shows you the granules that are stored in this one. Okay, phosphate granules. Yeah, magnetic particles like iron, gold, silver. Okay. So yeah, inclusions and granules are really important. That's kind of like their reserve. Okay, but not all of them are going to have it. Okay, we good so far? Yeah.
What is the G and H?
The G is for glycogen. H is for hydrobutyrate. Yeah. I don't expect you to know the whole name, but you should know PHB. Poly B hydrobutyrate. Okay, let's look at some endospores. You did this last week in lab, right? Yes, with the yep, to flame the endospores, and they turned out green, I hope. Did you find them?
Mm-hmm.
Oh, good. All right. So let's talk a little bit about endospores. They are really tough, heat-resistant cells. They're metabolically dormant. So if you happen to have endospores in your system, oftentimes it's not a target of antibiotics because you're not metabolically doing anything. Okay? You see them in only gram-positive cells. Okay? Like Clostridium and Bacillus. Okay? Yeah, so you've got two forms. You've got a vegetative form, and you've got the endospore form. Okay? What else? Oh, sporulation, the formation of endospores, and then when germination, when it returns back to vegetative growth. Okay? This is really the toughest of all life forms. It can withstand freezing, survives heat drying, radiation, chemicals. Okay? This is not reproduction. What do I mean by it's not reproduction? That if you have one cell, it makes an endospore. When it comes back, you still have one cell. It's not you're going from one cell to two cells. You're not doing binary fission. Does that make sense? Okay, so you have a cell, we'll go through the process of how you make an endospore shortly, but you have one endospore, it makes it, when it becomes, goes back into germination, it's still one cell, not two, okay? All right, so let's talk about how you make spores. Did you talk about this in lab?
Yeah.
A little bit? Okay, so here's the cell, and it's come on some hard times, all right? Can't seem to have enough nutrients of whatever it needs. So what it does is it makes a spore, and then this becomes the endospore. And you get the cortex and the outer layers, and then you get this mature endospore, and then this can be released, all right? And then when the environment is better, it germinates and it comes back. So it's not for reproduction, it's for survival, right? This is a survival mechanism, not a reproduction mechanism, okay? So what do you know about the bacteria that you were able to see endospores in? Were they stressed or were they living in Palm Springs?
Stressed.
They were stressed, yes. That's why it's always kind of sketchy, you know, are we gonna have endospores? Did we stress them enough? You know, that kind of thing. So, okay, any questions on sporogenesis? All right, so these endospores are super tough. They have resistance linked to calcium 2+, okay? And let's see, what else we have to know about endospores? Okay, so diploconate, okay, diploconate acid, and they're dehydrated, metabolically inactive. They can survive for 250 million years, okay? And they're only destroyed by pressurized steam at 120 degrees for 20 to 30 minutes. What process is that called? Anybody know? That's the autoclave. Yeah, that's why we autoclave everything, because we want to make sure that we destroy those endospores. Okay, any questions on endospores before we get to the medical significance of all this? Okay, so when you think about endospores, think about anthrax, Clostridium, Bacillus anthracis, and Clostridium. Clostridium tetani, botulinum, and perfringens. Okay, for Gascon green. That's where you see these. Okay, now we've talked about anthrax before, that you know it's been weaponized so you can inhale it, but where is it mostly found? Where are you going to find anthrax? You're going to find it in the soil, right? We talked about that. It's going to be in the soil. And animals tend to have it. So you see them, anthrax, sometimes you'll see cutaneous anthrax in sheep shearers, because the sheep's wool is in contact with the soil, and you'll get endospores in there, okay? But we can usually treat that pretty well. Unlike respiratory anthrax, by the time we figure it out, you're usually not gonna survive. Okay, tetanus, botulinum, and perfringens. So yeah, so those are the ones, when you think of endospores, those two should pop up right away, is Bacillus and Clostridium. Okay, is that good? All right, so there's a lot of different ways to classify prokaryotes. We can do the phenotypic analysis. What does phenotype mean? What is that? What you see, the things that you see, right? Okay, and then there's serological analysis where we're looking at antibodies or particular antigens. So we use immunofluorescence or ELISA. We have genetic analysis. The book talks about this too, the G plus C nucleotide composition. PCR, what does PCR stand for?
Polymerase chain reaction.
Polymerase chain reaction. What does it do?
It replicates. You replicate a certain gene sequence.
Yeah, you're replicating DNA, you're copying DNA. Okay, so genetic probes, microarrays, we'll talk about this later when we do microbial genetics. And then we do pathological methods, what phages. What is a phage? What is a phage?
Isn't it a white blood cell?
No. This is a mugshot of a phage. It's a virus that attacks bacteria. Okay? So you've got genetic material here, and then what is this made of out on the outside? Protein, right? And then it will, sit its butt down on top of the microbe and inject the genetic material in. This is what we call a phage. This is a particular one called a T4 phage. Okay, and this T4 phage, that's one of my favorite ones because it looks like a spaceship, doesn't it? Yeah, it comes down here. So there are certain phages that will only infect certain microbes. So that's why we can use it. And oftentimes if they have this, it can actually enhance toxicity. For example, cholera. If cholera has a specific T4 infection, the cholera will be much worse. Okay? So we'll talk about that a little bit later when we do chapter 6. But, okay, so we've talked about Burgess' Manual of Bacteriology, okay? And we've got domain and kingdom bacteria, all right? Also called eubacteria, consists of a lot of different phyla. So that just gives you some of the different phyla. Do you have to know the phylum? No. But you're gonna see some of these words, and so you're gonna be saying, oh, that's the phyla, okay? So let's talk about the bacteria that really are unusual. Mycoplasma, okay? No cell wall, okay? And it has a really tough membrane made of sterols, so it doesn't lyse. But that means that it makes it hard to treat, because if it had a cell wall, we could give it penicillin. And then otherwise, we've got to get antibiotics in there to treat it, but it's got this tough wall of sterols. What kind of biological molecule does sterol belong to? Lipids, yeah, lipids, okay? So this makes Mycoplasma pneumonia, which has been circulating around the globe since October. We saw a big spike in China, and it's just been going around. And a lot of folks had it over the winter. It's not your pneumonia that's like gonna kill you, usually. Mycoplasma pneumonia, they call it walking pneumonia, okay? Because usually you can keep up on what you're doing, but you still have it. So it is atypical, okay. Chlamydia, oh, a nice little STI, okay. These are obligate intracellular parasites, okay. And this is the most common STD there is chlamydia. Now, if you give birth and you've got chlamydia in your system, you get a severe eye infection for sure. Sometimes you see patients that have a chlamydia infection in their eye, but they're older. But we see this in babies that are born to mothers that have an STI that's undiagnosed. And oftentimes they screen for this before birth, okay? And then we've got this tiny gram-negative bacteria that is an obligate intracellular. What does that mean? Obligate means what? Must be, right? Must be. Intracellular, where is it going to be? Inside the cell, okay? And we see these rickettsias, Rocky Mountain spotted fever is caused, and oftentimes it's ticks. We see it with fleas and lice, but mostly with ticks, okay? So these are kind of unusual bacteria. So these infections, like rickettsias, oftentimes hard to treat. Why? Because that bacteria is hiding out in the host cell. That's pretty sneaky. So, okay. Good so far? Okay. A few more unusual ones. Photosynthetic bacteria. These are not medically important, but you should know that they're around. They sequester very unusual things. We have gliding fruiting bacteria that actually, to me, look at this. They're on a stalk. What does that remind you of? Funguses, yeah. Very strange. Okay, and then of course the archaea. These are extremophiles, extreme heat, salt pressure, all those. And again, not medically important because, you know, our bodies are pretty regulated as far as our environment goes. So, yeah, not gonna find them there. So, let's see how we do. Oh, that time. Okay, any questions on chapter 4? Good? All right, cool. Well, let's move on to chapter 5 then. Yay. Okay. 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 I put a bunch of stuff on chapter 5. Okay. Oh, by the way, did it help to write questions? Cool. Okay. Good. Is that something we should continue?
Yes.
Okay. All right. Cool. Now, I got to be honest with you. Some questions were like, wow. Wow. And some were kind of AI generated. I can tell those right away. Okay. I know that. All right. Please try not to do that for when you're trying to get points. I'd rather that you think of it yourself. If you're going to study using AI, that's cool, but when you're turning in something, it needs to be your own work. Okay? All right, is that cool? Okay. And some folks that submitted questions were Bio 1 level, not micro level. Okay? All right? But you'll have the first exam, you'll kind of see where things are. But you know, what's different from prokaryotes and eukaryotes? Oh, we're so far from that. Okay. What's the cell wall made of? Okay, do you know this? I don't know if you know this. Okay. There's something called have you ever heard of this, Bloom's taxonomy? Oh, I forgot to know. Taxonomy, me. Have you heard of this? Okay, some of you have. If you've ever done any education, you have. Okay, it's kind of like a little pyramid here. Okay, and this is knowledge, comprehension, and then we've got the ones up here and things like analysis, synthesis, and the last one up here is evaluation. Okay, so down here are 2 questions. Do you know, do you understand? Okay, your high school was like that. What is this? What is that? Define this, define that. Okay, a lot of Bio 1 is that. All right, but now you've got your foundation, we can work up here with it, which is analysis, synthesis, and evaluation. Okay, how would you compare? You know, these are compare and contrast questions. These are questions, like at the end of the semester, you're going to be doing an unknown. Okay, is that down here or is this up here? It's up here, okay? All right, we're working on the foundation here. Okay, and of course I'm going to ask you questions like that, but we want to get you working up in these areas as well, okay? And so a question like, what's the difference between gram-negative and gram-positive? Where does that belong? Down here. Okay, if I asked which cell is more permeable to lipids, what kind of question is that? It'd be up here, right? Because now you got to know lipids, you got to know the plasma membrane, you got to know if lipids will go through the cell wall, okay? You got to think a little more, okay? And some students say, oh, that's tricky. No, thinking is not tricky, okay? Thinking is thinking. If I just decided to ask you questions about things we never done, that is tricky. But thinking and putting things together, that's thinking. That's what is needed. Does that make sense? So when we do this exercise next time, I'll put some stuff up about Bloom's taxonomy, okay? And you can practice writing those questions, because your exam is going to be this, but it's also going to be some of this too, okay? And that's where the real fun starts, right? That's where the real fun is. I mean, when you're analyzing a patient that comes in, which level are you using? The top level, but you damn well better have this, right? You better have your foundation, because if you don't have your foundation, can you do this? No. So that's why, okay? So it's not only just the material, but we're trying to get you to think about it, and some of you are doing an excellent job. So is that cool? Okay, all right, good. So, got a lot of things on here. All right, so there's a video here from, this is Gokul. He actually took micro, he's sitting where you are. He took Bio 1, and he went on to be faculty at Harvard Medical School. And then now he's at Stanford. And this tells you about what he did. He's pretty amazing. I talked to him last week because he's got a student that we wanted to talk about. So anyway, 2005. So he was thinking, oh, I'm going to go and do this and this and this. He ended up transferring to UC Irvine, graduated in biology, got his PhD from UC Riverside in engineering, and then went on and developed this amazing microscopy technique that lets you see things in real time, and that has never been done. So I mean, he's one of these folks that sat in your seat and changed the world, just like you will. Okay. Let's see. Let's look at this one. This is pretty cool. Have you seen this?
No, I've seen that.
Yeah. Okay. Let's make sure we have no sound. This sucks. Oh yeah, we do. Right on. Okay. What? What? Oh man. What the heck happened? Okay. Okay, there's. Oh.
I took two things that I love,
Oh my
dancing and
gosh.
dogs, and I made them into
This is
a school
sucking.
business.
Okay. All right, let's try this. Okay, let's try it again. Oh, this is much better. No! This is not better. No, I know these, so it's like, okay. There we go. Now we're in the right place. All going on inside of you right now. many have seen that before? Raise your hand if you've seen it before. Okay, so a few of you have seen it. Okay, so how did this get started? This got started with, put you off, there we go, from Harvard BioVision. So what they did is they took all the microscopic data that they had and things that they knew about proteins and that, and they assembled it to make it so you could see it in action. Okay, so tell me, where are we right now? We're a blood vessel. What are these red things? What are the white things? White blood cells. Okay, cool. All right, now, you've heard about cellular communication, right? So this is some of the proteins that are communicating with one cell versus the other. Okay? Now, what are they communicating? Could be a variety of things. Oh my gosh, these are so cool. What is this? It's called a lipid raft. It's called a lipid raft. And what is it used? It's used for transport. We know in Parkinson's patients, they lose the ability to make these lipid rafts. How does that translate to the disease? Not quite sure yet. Okay, but you can see lipid rafts here. And you see all this communication, right? All these communication that's going on here. And with all these molecules floating around. Okay, now let's look at some other things here. We're going to back off here. Okay, what the heck is this? That's part of the cytoskeleton. That's the scaffolding. Remember you learned about the cytoskeleton, about intermediate filaments, microfilaments, and what else? Microtubules. Okay, this is Bio 1. Okay, so you're looking at the scaffolding. That's there. Mostly microfilaments and intermediate filaments. Here's assembly of a microfilament. Okay. And we know microfilaments are made of actin and myosin and that, okay? And here's an enzyme that's going to break it. Boom, okay? So we've got ways. Oh, what is this? What is this one? It's the biggest one. Which one is it? Yeah, microtubule, right. Very common microtubule assembly, disassembly. When do you see microtubules? What part of the cell, eukaryotic cell division, do you see it? Microtubules, you see it in mitosis. Okay, from a medical standpoint, there are several chemotherapy agents called Taxols that target microtubules. Okay, what's going on here?
That's a kinase, isn't it?
What is it?
Kinase?
No, not quite. This is a motor protein. And it requires ATP to do this, and it's carrying a vesicle. And see all these dark things on it? What is it doing? These are signaling molecules. So we're not sure what's in that vesicle, but it's got a bunch of signals on it. And look how it moves across that microtubule. Okay? This is the best shot here. What's this up here? Mitochondria. What's down here? The nucleus. And if you look really carefully, you can see these two things that look like two pieces of licorice that's used in cell division. What are those? Centrioles. Oh, and back here, what is that? What is that right back here? The Golgi apparatus. What does the Golgi do? It ships and packages what? Proteins. Good. Okay. What membrane is this? No, it's a nuclear membrane. Okay? And we've got some RNA, and here's the ribosome assembly. Okay, boom. So here's our new protein, here's the ribosome. Okay, there's the gorgeous mitochondria. Ooh. Okay, now what is it doing? Well, it's there's the RNA, the new protein's going back into the nucleus. Okay, what are these? Those are little vesicles off the Golgi. Aha, yeah. Gorgeous. Okay. Now, we've got, let me back up a little bit. I went too fast through there. Okay. So now, what process is this?
Exocytosis.
Exocytosis, yeah. Okay. And now we're making another lipid raft so we can move things. Okay. Again, we've got cellular communication going on here, linking, and this is a very special process that happens when a red blood cell gets out of the capillary. What is it called? Does anybody remember what it called? It starts with a D. Diapedesis. Have you had all this in anatomy in Bio 1? You've seen all this. This is it in action. Okay? And when I'm thinking about this, I'm thinking, well, that's great. That's a healthy cell. What do we look at? We look at cells that may be diseased or things like that. So, what I did is I put some more videos on here and some reading. So, because you've done eukaryotic cells in Bio 1, in 50A, in 50B, you know the healthy ones. This is to help you so you can chunk it a little bit, okay? So you can remember what all of these things are, because really we're just looking at cells and diversity here. So let's take a look. Here we go. All right, here we are with eukaryotic microorganisms. Let's look at this. This is kind of cool. Okay, you should recognize some of these on here. These are all structures that you should be familiar with, right? What's the role of the mitochondria? What happens there? Okay, cellular respiration, not the powerhouse of the cell. That's a Bio 1 answer. You're in micro. Okay? And we know what this membrane is. What is this membrane? It's the intermitochondrial membrane. What magic happens there?
Electron transport chain.
Good, the electron transport chain, good. And inside here is the matrix. What happens there? Oh, that's test material. What happens there in the matrix of the mitochondria? Better go back. Look at chapter 8. Okay, that is where Krebs cycle happens, right? Where does glycolysis happen?
In
The cytoplasm. Very good. We saw the Golgi. What is this? Right here. The rough ER, and it's rough because ribosomes. So you know you've got protein synthesis here. Okay, smooth ER. What does a smooth ER make?
Lipids.
Lipids. It's a site of lipid synthesis. Okay, you see, what's the trash that takes the trash out? Peroxisomes and lysosomes. What do? They digest things. Lysosomes with enzymes, peroxisomes with peroxide. Okay, you're going to run into lysosomes when you see liver liver disease. Thinking of a liver disease. Alcoholic hepatitis, right? When you get all those ascites in that, it's usually because the lysosomes you need so much lysosomes because you've got so much alcohol, and the dehydrogenase isn't taking care of it, and then it starts digesting the liver. Yeah, good times. Not really. Okay, this is a weird cell because do you notice something that doesn't belong there? What doesn't belong there? Yeah, the chloroplast. Where are you going to find that? Plants. I don't know who did this. It was silly, so I like it. Okay. What are the 3 things that plants have that animal cells don't? That big central vacuole. Cell wall made of cellulose. Good. Okay, so good. If these are, you're like, oh, I'm not so sure about this, bone up on your cells. Okay, you've been through at least one class that did this in detail. Okay, we've talked about why organelles or ribosomes are not organelles. Okay, let's talk about the origin of eukaryotic cells. Okay, there's something called the theory of endosymbiosis, or the endosymbiotic theory. Okay, where here's a cell, a big cell, here's a little cell, and the big cell engulfs the little cell, and then whenever it goes through mitosis, the cytoplasm divides, right? So you get this cell, and eventually, what are these? These are organelles. Kind of cool. So the endosymbiont theory, that kind of, is the theory that we're working on right now, or at least we're operating with, that these organelles used to be little tiny cells. Okay, we've talked about cilia and flagella, right? Cilia, how does it move? How does flagella move? Okay, all right. And here's the microtubules. We just saw those. It has a 9+2 structure. So you've got 9 pairs on the outside, 2 in the middle. Okay, so got cilia and flagella. That's pretty neat. Where are some places you have cilia?
Upper respiratory.
Upper respiratory tract. Good. Okay. Let's see. What else we got here? The nucleus. Oh, it's 52. Yeah, 3 minutes. Okay, the nucleus, membrane bound, double helix, histones, so you can compact this 3 meters of DNA per cell. Okay. Rough ER, what is that? We talked about that. That's where protein synthesis occurs. All right. So we also know about the endomembrane system, right? Where things are able to move. You get proteins made, goes to the Golgi. The Golgi can either secrete it or it can stay inside the cell. Okay, so remember the endomembrane system. Okay. Mitochondria and chloroplast. Which one is which? Okay, here's the mitochondria, and guess what? It's got its own DNA. And then the chloroplasts, and you've got stacks of granum in the individual thylakoids. Okay, same 70S ribosomes. Okay, and then we've got cell division, right? Complex cell division, mitosis. All right, have you been through mitosis? What are the stages?
Prophase, metaphase, anaphase, telophase, and then cytid.
Okay, which phase do they line up in the middle? Meta. Which one do the chromatids separate?
Anaphase.
Anaphase. When can you start seeing chromosomes? Prophase, right? Prophase. And what is? Do you remember the cell cycle? MG1, SG2. Yeah, remember MG1, SG2. Okay. All right, I'm going to stop there for today. All right, chapter 5 is not on the exam. It's not on your nudge sheet. So anyway, yeah, that's for the next one. What questions do you have?